get started.
Cool.
Good afternoon. It's my great pleasure to welcome you to the third installment of the CALM BIMR seminar series. And today we're very fortunate to have Dr. Joshua Koenig. And so Joshua did his Bachelor of Health Sciences here at McMaster in 2015, and then a PhD under the supervision of Dr. Manel Jordana, working on the generation of high-avidity IgE antibodies for food allergy. And so within this area of research, he has continued developing applications for immunology and for intestine and gut health. And he is now a research associate at the Schroeder Institute for Allergy and Immunology, and also the associate director for that institute. So it gives me great pleasure to welcome Josh, and he'll be talking about highly multiplexed imaging to capture biological complexities. He's doing at CALM.
Yes, I am. And you'll hear those words a lot of times in this. To start off, I mean, this is a pretty diverse crowd. I'm an immunologist. I study the immune system. And, you know, I kind of belong in two places, in the Schroeder Institute and in the Immunology Research Institute, or Center, sorry. But you know, in asking myself, what can I say to a materials research institute, I, you know, was, you know, kind of noodling on that and trying to keep it at a level that makes sense. But I've been assured by my good friends, Jose and Joel, that I am in fact not an immunologist. I am a soft biomaterials scientist. You know, a lot of the work that we've done is at CALM, and you'll see that logo a lot here, because it's really a part of our plan as we move forward. The issue at hand at a very base level is that biological systems are highly complex systems, and we go very reductionist in trying to study these systems, and that's not really the best way to do it in many cases. This is a picture of the small intestine, and you can appreciate that there's lines of cells that separate the inside of the intestine from the base of the intestine. All this, you know, light, I guess, peach color is the us side of the intestine. There's many different cell types along here, from epithelial cells to cells that make mucus and all kinds of other products. And underneath that, there's all the cells that I like to talk about and think about, which are immune cells. And these cells are all working together to create a tissue environment. And when I say that this is very complex, when you're trying to identify those cells, the way that we've started doing this, or not we, but immunology as a whole, is by looking at the things that these different cells make and using that to define them. So these are labels or markers that we use to say, this is what this cell type is, and that's what that cell type is. But there can be many markers. I mean, if there's any immunologists that are listening, they'll recognize some of these molecules and realize it's ridiculous to put them all in the same cell because some of those are mutually exclusive. But the constellation of what markers a cell does express or what markers a cell doesn't express is really what defines a lot of the function of the immune system. And I wanted to give you a small example of the biological complexity that we're thinking about to kind of set the stage for why we're doing the stuff that we're doing. So I study, my group and the Allergy Institute as a whole, we study primarily food allergies. And in case you were wondering about how food allergies actually happen, we have these antibodies called IgE antibodies that are floating around in all of us, or well, not all of us, but in allergic people. Non-allergic people don't have these. And when a peanut antigen is experienced, when you accidentally ate it, or actually there's peanut and dust everywhere. It's crazy. I don't know how there's peanut everywhere, but there's peanut everywhere. And if you're exposed to that peanut, what happens is that these IgE antibodies are on the surface of certain cells called mast cells. And when they're triggered by peanut, it causes these mediators that makes you have a fast reaction. These anaphylactic reactions within minutes after they've eaten a food. And so these IgE antibodies are really good to study. And if I know anything, I think I would say that I know about IgE antibodies more than even allergy. These IgE antibodies come from a different kind of cell, which we call a plasma cell. And really their only job is to make antibodies. But the conundrum of this whole situation is that the cells that make these IgE antibodies, they don't live for very long. And so what we've come to learn, and this is work from my PhD mentor Manel Jordana's group, which I still obviously work with, we've learned that memory B cells, these quiet cells that stay in all of our tissues, are the ones that end up making more IgE antibodies. And so we asked ourselves a pretty simple question in terms of biological complexity, which is there a type of memory B cell that actually does this IgE production? A pretty basic and fundamental question. And so the way we went about trying to deal with this complexity is we got a bunch of people and we took their blood, and from their blood, we took memory B cells. And we used a technology called single cell RNA sequencing, which takes the protein encoding RNA or mRNA from every single one of those cells and sequences it separately so that we can say what each cell is based on the products that it can make. And we can do this in a high throughput way. And so when we did this with 90,000 cells from allergic and allergic people, we found incredible diversity within the memory B cell repertoire that was not really appreciated. People would call memory B cells a pretty homogeneous group of cells that didn't really have different functions or different subgroups. There are some reported in the literature, but nowhere near the 21 different clusters that we identified. And so what you're visualizing here, every single dot is a cell in this context. And we've used statistical tools to cluster these cells together based on their likeness, how similar their expression patterns are. So we know now that there's 21 different clusters of these memory B cells, and trying to understand that diversity is extremely important. When we take a look at the ones that are making this IgE molecule, the one that I was telling you about, we have an incredible homogeny within this one population, this cluster 12 here. And we have a paper that we've just submitted that is describing this population, a new population, and describing its functions. And we have evidence directly in humans that these are the cells that are making IgE. But that same complexity that I was just telling you about exists in every single tissue in our body, every single cell type. T cells have that kind of complexity, macrophages, even our structural cells in our body. And knowing that that diversity exists, so saying there's 21 different kinds of memory B cells is really not enough to do anything with. How does that actually tell us what's going on in this disease? We know these things are associated, but how? What are they doing? So, you know, looking here, I kind of sneakily hid a few little Easter eggs in there that I've now highlighted for you, which is that there's a bunch of cells that are talking to each other here that are beside each other. And the reason I put these in here is because the immune system is a system of interactions. It's two different cells talking to one another that results in some output. We have these T cells that are known as pretty much the master regulators of the immune system that go and tell other cells what to do. And we know that those are very heterogeneous. There's, you know, every year there's a new paper saying there's a new subset of these things. We know there's 21 clusters of memory B cells. Who are these 21 clusters of memory B cells interacting with? And what is the outcome of those interactions? These are the types of questions that we're asking ourselves now. And we're trying to figure out how to do this. So one way to do it is to look in situ. And I think that this is really the right way to do this. And so this here is a pretty early image that was captured. Actually, I think I did this one. But the way that we try to do this is by staining tissues, sections of tissues that we've collected from either humans or from mice, and staining them with a couple different markers here. These are different proteins that are expressed by these cells to explain some of this complexity. And you can see that within this one tissue here, which this is a lymph node, there's regions where the green cells live and there's regions where the yellow cells live, and those are different regions, but there's also yellow cells up here and yellow cells down here, yellow cells that are near red cells and yellow cells that are near green cells. And you can hear that it becomes a Dr. Seuss story over time. There's all these different places these cells can live and different things that these cells are interacting with. And the question is, how do those interactions result in disease, or how do they result in, you know, the opposite of disease, cure, in the hope of a biologist to cure a disease, for example? So we're trying to understand the localization and interactions that the different cells of the immune system has. And our hope is, in looking at these things in situ, that we can come up with new interactions that people don't know about, because we have a habit of taking the cells out of their tissues, taking the cells out of their tissues to study them using single cell sequencing, like I showed you in that previous slide. So if you're going to do microscopy, the difficulty is we have to capture tons of complexity, and the limitation really ends up being how much complexity can we look at on one tissue? And if you talk to most immunologists and biologists, the exception maybe being Ray Truant in this room and other people who do some good microscopy around here, most biologists will really only use 4 colors for their microscopy, which is the equivalent of this picture that I showed you here. Really not particularly complex, and I can't divide these populations. The way that they do this is, we have 4 laser lines and we use one label per laser line, and you get violet, green, yellow, and red marker, basically. So we can look at 4 things at one time. This is clearly not enough. So, if you look at newer confocal microscopes, and, I think a lot of biologists are actually stuck back here. Do more than 4, it's the only thing that you can possibly do. And that's not true at all. In fact, microscopes that we have in CALM can do far more than that. And there are good microscopes at McMaster that can, with relative ease, get you up to 6 or 8 color imaging without really trying very hard. I mean, you can look at a 405 nanometer laser and you can put 2 colors, which if anybody in the audience online or here is an immunologist, you might recognize these labels or these fluorophores that I've included here. These are very common reagents, flow cytometry reagents that you can purchase at any major distributor, right? With just very common labels, you can generate 1, 2, 3, 4, 5, 6, 7, 8 color microscopy images. And again, this isn't particularly difficult. And I know because we've done this many times and we're doing it routinely. There's some considerations you have to make. These things are done a little bit more easily with tunable detectors to be able to decide what light you're going to accept versus not accept. You might have to use some clever sequential scanning to make sure that you're not seeing overlap. And even if you do see overlap, all of the major microscope providers these days have computational tools to unmix colors. So if there's overlap in one channel that goes into another, you can create basically a matrix and compensate out those overlaps. And it's relatively simple. So we've done these with a lot of great success. Jake Colati we'll talk about him in a minute, but Vittoria Lintho, who's sitting there, has done many 6 or 8 color images over time. It's actually funny because I just received an email from Reg, who's sitting over there, who says that we are doing 11 colors frequently now on their Leica systems. I didn't know that you were going to be here, Reg. Hi. And I figured that looks something kind of like this, the exact same setup that I showed you, but we're now starting to move into the NIR and starting to add a couple labels out there. So this is Alexa Floor 780, and this is Alexa Floor 800, 750 and 790, I think. And then we need one more label. I imagine that there's probably a very far red label that you could use, or really the reagents that are coming out now are coming out so fast because multiplexing is such a big thing that we're getting longer Stokes shift dyes that we can use on the 405 laser and have them be detected out in the 700s, for example. So there's ways that you can get up to 11 colors. And why I'm telling this to this audience here is because if you are a biologist who has come to this talk and study the complexity of the tissue that you want to study using microscopes that exist today at CALM or at TARI. And you can reach out to the people at CALM, you can reach out to me, and we can help facilitate that for you because this is not particularly difficult. However, small advertisement break, we're starting to go even further than that. So I told Joao and Mohamed and Jose that they'd see themselves a lot in this because they've had a big impact on us. But CALM is looking to get a new microscope designed for multiplexed imaging, exactly like what I'm talking about today. So I put CALM in a very big square here because this is mostly led by them, and I put us in a very little small square here because we made a small nominal contribution to this as well. But there are many laser lines on whatever microscope we're going to get. I don't think I'm allowed to say where we're leaning, but whatever microscope we're allowed to get. There's detection that goes from 400 all the way to the 900 range. It can be fast. You can get simultaneous detection, and you will be able to do an 11 color image with the tools that we have here at McMaster. So the problem is that 11 colors is not 21 clusters. That's the issue. We can't even study the complexity that we see within our memory B cells with what I've just told you. So, if I just wanted to look at the memory B cells, I would need 21 colors. But if I wanted to see the other cell types that are in this tissue, this complexity that is in this tissue, I would need to have, up to 30 different markers for 30 potential different cells that are in here. So now we're looking at 50 markers, which we're really not going to be able to get, or, technology would have to advance a lot to do this in a single pass. So if I wanted to add other markers like question-specific markers or functional molecules, you can see how this would ramp up very quickly into a very high multiplex situation. And for us, that is where, well, in probably just about November 11th of 2020, I saw a paper that came across my pub crawler because of one of the names that was in this. And it's this new tool called Ibex, which is a multiplexed imaging tool that allows you to study many markers in complex tissues. And the way that this tool works is that you can with your set of 6 or 8 or 11 dyes. You can go and use one of the great microscopes at CALM to detect those 6, 8, 11 dyes, whatever it is that you're doing. And then you can inactivate those fluorophores using a bleaching reed, lithium borohydride, which will reduce the fluorophores that are existing there, with the exception of a few fiducial markers that will stay. And then you can go back and stain with the exact same set of fluorescent molecules and do it again. And you can do this theoretically infinitely. So you can keep going and adding more labels and more labels and more labels and aligning those images to a point where you can have very high multiplicity tissues. And so in practice, our collaborators have done 68 markers on a single lymph node by comparison to the regular 11 that we would be, looking at the higher end in today's microscopy. And in theory, this is infinitely scalable. And I know Andrea Radke very, very well after a year and a half of collaboration. The reason they stopped at 68 is because they ran out of markers, not because they ran out of interest or ran out of ability to do it. They just didn't have more antibodies in their fridge at the time. That would be different now, but back then they just didn't have more. So, Josh Koenig in 2020 thinks to himself, I can do that, so I should. And that was a really interesting thing to think, because at that time I had never used a microscope before. So I figure, let's go to the Olympics before I learn how to run. And so because of these insane ideas that I'm spouting at you, I'll give you a little bit of a history of our imaging trajectory, our journey that we've gone through. So remember that in November of 2020, I thought I can do that. And then in January 1st of 2021, we recruited this nice guy, Jake Colati, who is really an incredible scientist and drove us very far in our Ibex journey. Over the first month, we had many failures, and that was primarily because of my ineptitude of knowing how to do any imaging whatsoever. We were trying to fix cells with acetone, and we were destroying fluorophores and losing tissues, and it was a mess. But it's always nice to show your first success. So this was our first image in the lab. It was our first one-color image that we had on a wide field Zeiss microscope. That is DAPI, which, if anybody knows DAPI, you can sprinkle that on the air above your sample and it will stain. So the fact that it took us a month to see DAPI is actually pretty embarrassing. That was our start. Around March, so at that time we only had access to a wide field microscope that was in the Immunology Research Center. And we kind of knew that we would have to go to some more high-powered confocal space to do this. And at the time, I had a TA by the name of Sarah in the course I was teaching, who said, well, over at Tari, which is at the General Hospital, we've just purchased a new microscope. It's a, front of the line, like a Stellaris microscope. Maybe we have access to that. And I said, sure, well, ask, the PI if we can. And, things worked out well, and we got access in March to this Stellaris. So now we can capture, higher multiplexing. So around that time, we got to 2 or 3 colors, which was quite nice. And we were doing this routinely for a little while. And then we figured out stop doing in terms of how we dealt with the tissues. We moved into doing some fixed frozen tissues and some very careful sucrose gradienting. And then we OCT embedded the tissues to basically keep as many antigens available as we could. And after a little bit of learning in June, we started to routinely collect 5 or 6 color confocal images. So if anybody here is sitting or listening as a biologist thinking, okay, how long will it take me to get these images? If you had absolutely no help and you were as bad as us, then it might take you about 6 months to do that. I can say, because you're here, do not do this. Come and talk to me or go and talk to CALM, because they will help you avoid all of our pitfalls. But it wasn't much longer after that Jake's persistence paid off. We tested nearly 80 markers of antibodies after that time over the summer in 2020. And then in August, we had our first Ibex image. So the trajectory of us getting a 5 or 6 color image to getting a multiplexed image was incredibly quick, because when you have the base microscopy tools down, the technology itself is not particularly difficult. And I'll talk to you a little bit about our strategy for Ibex, how we're doing things right now, which is that we are doing multiple rounds of imaging. You have to take the slide off the microscope, bleach it, restain it, and go and image again, and use a fiducial marker to align these things and make basically one image with many different colors. We have landed on using this, which we are probably going to update as we get a better microscope with a little bit more capability, but we are using DAPI as a fiducial marker for a couple reasons. The violet dyes are not particularly strong in detection, in our hands anyway. They don't bleach as well. They're actually very frustrating synthetic polymer molecules that don't like to be reduced, so they tend to stay fluorescent for a long time, and that's a bit of a pain for us. So we've swapped those out. We just use DAPI, and then we use this set of fluorophores. And I know that there are many people here who are sitting and thinking to themselves, why the heck does that say PE? And I have fought back and forth with Joao and Jose and Mohamed about this, and they know exactly why that says PE. PE is a horrible molecule that photobleaches immediately, and I know that. The reason is because almost every single reagent we use is a commercial reagent that you can purchase and have delivered to your lab intact, ready to use. We try to avoid, as best we can, having to make our own conjugates, because in our experience, all that results in is other people not being able to replicate our work. So I'm going to talk a little bit about the Ibex imaging community in a bit, and we can talk more about that. So this was our first Ibex image. We'll talk about quality of the imaging afterwards, but this was a 17-color image that was collected by Jake Koenig here. And this is of a lymph node studying a few different immune markers. And here I've kind of broken it out. These are all from different cycles of the Ibex imaging, but you can overlay them and they work perfectly together. And what is really interesting that we found about this, which again, I'm not going to stand by the quality of the microscopy here. We'll talk about that in a second. But we found a really interesting observation that there's tons of staining for a different type of antibody called IgM that exists right around the vessels in these lymph node tissues. And we have looked and looked and looked and looked and looked, and as far as I can see, nobody has reported. These are not cells that are making IgM. This is IgM that is depositing on the surface of other cells. When we look for the cells that make IgM, they're in different places. So it's almost as if biologically something is trying to restrict this IgM antibody to stay here and to not move around. We were able to show, because this is a zoom in this area, that a lot of these cells are CD11b expressing, which would make them things like macrophages or other myeloid cells. So they're capturing these complexes on their surface. And this was a surprise to us. And I think that this is, we've shown this to a few people, and it's been a surprise to many people. And I think the reason is that we just never study these things like this. So we never really thought to look. And I think that this is what the power of Ibex is going to be moving forward, where we're going to be able to find things that nobody has really seen, and it will be low-hanging fruit just because we don't do this in situ very often. So we want to follow up on this. The only problem is we don't know how, because I don't study IgM and I know nothing about it. So I need to find some collaborators who know a little bit more about this antibody, who can help us know what it is that we need to do next in this domain. So this was very exciting to us. It was our first test at this, but the question is, well, how do you analyze this data aside from looking at it? And then the answer is that we can pretty well just turn it into the exact type of flow cytometry or dissociated data that we would normally have using computational tools. So we can put a segmentation mask using a variety of different tools on top of this image to segment out all the cells. And then for each cell using MATLAB, we can determine the average expression of every single marker in every single one of the images. So we basically have what is, the same as my single cell sequencing data. It's one cell and all the parameters that cell expresses. And then from that, we can apply clustering analyses. And this was the first one that I had done. And it ended up coming out very clearly in terms of the biology. I mean, if you take a look, this may be lost to many of the materials researchers, but if you look at the different populations, the clusters that are very similar to one another in biology are actually quite similar to one another on this plot as well. So in reality, this is working out quite nicely. And what you can do with this data is you can then go and recreate the image. So I've shown you the image here, and I've plotted the clustering information for the data, basically maintaining the position and taking the cluster that I got from our computation and overlaying it. And then I chose nice colors that match here, and you can see that our blood vessel is still going right through the center here. You can see that there's this red outline of our what are called subcapsular macrophages up at the top. And you can, by and large, recreate the map that you were seeing before and start to ask questions about these cells. And the types of questions that you could ask are, for example, which clusters are near each other in a statistically significant way. So you can look at every single cell in a cluster and measure its distance to all the other clusters and see on average which ones are closer to one another. And that gives you an insight about the biology. What's happening? Are these cells talking to one another? You can, for us, we can ask whether or not our memory B cells, our different types, are preferentially located in different places within a tissue. You can either use AI to determine the places in the tissue that are unique, or if you know your tissue, you can just draw circles and then count how many are in there, obviously computationally, because I'm not going to count that many cells by hand. The other thing that you can ask is maybe, hey, maybe it's not other cells that these cells are touching. Maybe it's a certain composition of matrix that makes those cells stay there. So you can stain for the extracellular matrix and then take every cell in that cluster and determine just on average what markers are around it in that area. And then lastly, the jury's still kind of out on how to analyze this data because these tools are so new. So we're really open-minded about how we can do different analyses. And to that end, we've have a very young collaboration with Dr. Andrew MacArthur, who's a computational person. And when I say very young, we've agreed that we want to do stuff. We just don't know how, what, or why. So we will figure those things out. But we know we want to do things because we both like this stuff. So I want to show you a little bit where we've gone. Now I've put Joao's face up here because Joao, really helped us learn a lot more about microscopy and a lot more about how to get better images. So to put into perspective, this is what we were seeing before, which is a little bit of a mess. And Joao helped us collect these images during demos that we were doing as we're selecting the microscope. And so thank you very much, Joao. And these are images that were captured by Vittoria, again, who's sitting right there. Vittoria collected 5 rounds, is that what it was? Yep, so she's nodding, good. And what you're seeing here is that because Joao helped us figure out our microscopy stuff, we were able to capture a much larger field of view and look at many more cells in these contexts. So this is a huge field of view, and these here are 2 different imaging rounds, and they look pretty well identical. And when you zoom in, you can see here that these is the exact same area on the tissue, and you can almost envision in the middle here, the vessel that's going up that is stained in this green color right here. And I'm realizing as I'm showing this that Vittoria is right. I need to blow these images out a little bit more so people can see them. I always disagree with her because I don't like blown out pixels, but she's right and I'm wrong. What you're seeing here is a whole bunch of different cells that are expressing these different 3-color images. So there's some co-expression that you can see if you look closely between these 2 different cycles. And then there's many cells that are differentially expressed that have some of the markers but not others. But the thing that really keeps me up at night is the dark space. What is living in those places that we haven't actually put a label yet? And that's where increasing our multiplexing will help us because 1, we can put on dyes that will help look at all the membranes. It's something that we've been working on. And then we can know that there's a cell there, but we can't see what it's expressing. And that's the position that I don't want to be in. I'd prefer to have a label for every single one of those cells so that when we find our memory B cells or when we find these different cells, we can pinpoint exactly what they're communicating with in statistical space over many thousands of cells. So this was a really beautiful image. And I'm very happy to show it. So thank you, Vittoria. So what is our goal? Where are we trying to go? And I hope that this is working. This is work that was done by our collaborator, Andrea Radke, that was published in PNAS when they described Ibex. This is one of their lymph nodes where they did 66 parameters. And they actually did tiling of the whole lymph node. And this is a human lymph node. And you can see that I haven't quite figured out how to do just yet, but when I figure it out, it'll be in my presentations. That you can see all these different regions of these different colors that are of the different markers that are expressed in these areas. And it's so clear that there are cells in here that are long, stringy cells, for example, that don't come out nicely when you crush cells up into a single cell suspension to try and figure out what's going on. So our hope is in using these more structural cells or the interactions with these structural cells and how those are actually working. I think that one of the best uses is to create highly multiplexed maps of many different tissues. And there's a few different consortia that are trying to address this, basically getting a tissue, kinds of different markers on it, and then understanding the complexity of that tissue. And so we're hoping to contribute to that. But the last thing is that multiplexing in Ibex is not just about getting as many markers on a tissue as you can. If you have a question that requires 14, not 11 markers, then you can use one round of Ibex imaging to get to that 14 marker point. So even smaller questions that don't involve tons of optimization, tons of markers, can be assisted through Ibex. So, I want to talk a little bit about the projects that we have in the lab. I asked these people for their pictures last night, so thank you everybody for sending them. Victoria's presently working on moving us into the human space, which basically means she did all this lovely work and then she has to get square one and fight with everything again. So she's a trooper, and I really appreciate that. This intestine work that I was just showing you, Aidan Gagnon, who's up here, sitting right here, is going to be taking on that and trying to expand upon our intestinal Ibex imaging to understand a little bit more about what T cells are doing in that space. And then I just threw this one up here because we very recently recruited Savin, who is also here sitting in the back corner. And Savin is going to be joining us as the first graduate student who's working on this multiplex imaging, which I should also say, everything that you've seen here has either been done by me or undergraduate students. And that kind of goes to show you the incredible talent that we have in our lab, but also how accessible this can be for your labs if you want to try and do these things. Although I will say, there's something special in the sauce that made Jake and Victoria, so you might not be able to do quite what they've done, but you can get close. And Savin is, I'm really excited, is going to take on this memory B cell question that we've been trying to answer, and he's going to, I'm sure, take this really far, and we're really excited to have him. So I want to point out that Ibex is not the only multiplexed imaging modality. There are many other imaging modalities, but I think Ibex is one of the stronger ones out there. So I'm going to start all the way over here. And the reason is you can, in the same way that you can replace flow cytometry with mass cytometry, basically using, I believe it's called time of flight spectrometry to look at very narrow peaks of the different metal ions to increase the number of markers you're looking at. You can do the exact same thing by imaging using something called a MIBI-TOF. And the reason that I wanted to bring that up is because we have a MIBI-scope here at McMaster that's run by Jonathan Bramson and Jamie McNichol, and they're both incredible people. And I think that this is a very complementary technology that should be used. I don't see it as a competitor at all. I think these are complementary tools. One thing that's great about MIBI-TOF is that it's a one-pass technology. You can go right through your tissue once. You don't have to do things cyclically. However, there is an upper limit of 40 markers that really haven't been surpassed in the metal space just yet. And that's something that Ibex allows us to go right through and as high as we want. The other thing is that the reagents to do this are extremely expensive because you either have to purchase metal conjugates or you have to make metal conjugates, which is a lot trickier than it sounds. There are DNA label technologies, which actually the folks at CALM have been asking me a lot about recently. I think that this is a good tool, but again, I think you have to create a lot of reagents for it, and I don't think that that's a great way to go about doing things. Instead, these tools here, of which I think Ibex is the strongest, allows you to use commercial reagents. Everything is available. Nothing is proprietary by any company. It's all stuff that you can either make or purchase and have delivered directly to your lab, which I think increases how replicable the stuff is, but also allows people from any country to be able to do these experiments. And then the other thing is that this is very modular and expandable, and it tends to be a little bit less expensive than these other options. So, these other options may be interesting to you, but for now, we're doing Ibex here. I want to talk just a little bit in the last few minutes that I have about, creating a global Ibex imaging community, which is something that matters a lot to me and Andrea Radke, who has created Ibex. We're trying to create a large group of people who can contribute to all the clones that they have used and tested, whether they've worked or whether they've not worked. And the way that we're doing this is we're committing to full open source. You can get everything that you need from a commercial distributor. You don't have to make anything new, and nothing will be hidden doors and nothing will be proprietary. All the protocols for doing Ibex imaging will be fully open and iterate. We'll use commercial reagents as often as we can and try to minimize cost by sharing as much as we can. What you'll find if you go here right now is nothing, because we're still trying to build it. But once it's done, is you'll be able to find protocols for how to do everything from grossing to fixing to embedding tissues, sectioning and slide preparation, how to block and stain, suggestions for imaging. That's a little bit harder because everyone will have different microscopes, and then bleaching and restaining protocols. You'll also find a massive repository of imaging reagents, things that have been used in all the conditions that they've been used in, and whether or not they've been used on your tissue, you'll be able to search for that. It'll have the tissue that they use, it'll have the staining information, buffers, everything, if any deviations from the protocol need to be made. And then the thing that we're working on now is how we'll be able to host validation information of how you know these tools are working the way that they're supposed to be working. They're targeting the right targets. And then the last thing we're going to try and do is create Ibex panels that are publishable. So we put together an Ibex panel, say this works for this tissue, we'll publish it, give it a DOI, and people could follow it directly as we set it, and it will work every time. So I also want to talk about building a local community of imaging here at McMaster. I think this is something that I'm not really even at the forefront of. I think that the Center for Advanced Light Microscopy is doing big imaging community here at McMaster a lot better than we're doing it, but I think we can contribute. The reason I wanted to put this up here is because if there's any HHS folks that are here, please go to CALM. I've been hearing it won't walk because it's too far away, which is insane to me, because even being in the same room as Joao and Mohamed and Jose, Ray, and Xian, you sense the things that you don't know and the things that they're going to be able to help you with. And you know that when you go in there, you're getting the best that microscopy really has to offer. And it is worth the money. It's worth the fee for service. And remembering paying those fees keeps those people, allows us to keep this work. And also, they're just great people. They're really fun to be around. I've had a great time. How I'm doing Ibex here is that I'm running something that I'm calling an open bench, which is literally this bench right here, which was the bench that I worked at during my PhD, and I've now mostly vacated. The idea is that, and do Ibex with us in MDCL. We have all the tools that you need for the imaging. Here I've shown some slides, the gelatin adhesive, and all the different stuff that you would need to do the imaging. And the idea is also that we have tons of reagents that we are using. And when you buy a vial of antibody, it's a big old vial. And we're not going to use all 100 milliliters or microliters of that big old vial. Other people can come and use that as well. We have over 100 mouse antibodies, and we're starting to bring in human antibodies thanks to work by Vittoria and Mike Kamal. And we're creating a kit. And the way that we're hoping that this will work is that you can come and use Ibex, and you'll get access to these things, to our protocols, to Vittoria, to Aiden, to other folks that work in the imaging, to me. And you'll have access to all of our reagents in our kit. You can give us, but you have to provide a student who's going to be dedicated to doing the imaging. So we have a lot of the reagents, but you need to give us the people who will live with us and can learn from us. You'll have to replace antibodies that you use very frequently. And then any new antibody specificities that you validate will stay with the kit, so we can continue expanding upon our open source platform here at McMaster. And then the last thing that we would ask is that you provide all of your validation data and everything to our global Ibex imaging community so that others can follow it as well. And then, this is Ibex, but I'm also very happy to support any tissue immunofluorescence. If you just want to do 6 markers or 8 markers, come and ask me questions, because it's almost certain that we can help you. If it's mouse, if it's human, let's talk in a month, no, a year. Vittoria can tell you when we can talk. This is working, I would say, and I think it's working quite well. We have quite a few collaborators now, and it's growing, pretty frequently. So, I put us here, these two, but from the Farncombe family, you'll see on the last slide, IIDR and Andrew MacArthur is getting involved. People over at Firestone, like Megan Veerhout from Shuttle Ask's lab, is doing some big Ibex work. And then we have people from what Mike tells me is called EMRG, and I had not heard of this before, and that's my mistake. But Mike Kamal from Johnny Parise's group is looking to do Ibex on human muscle cells, which is very, very exciting. So we're also looking to build collaborations outside of this biology space. We want to try and automate this Ibex workflow using microfluidics, and we need engineers to help us be able to do that. We think that there's probably interesting non-biological applications for this, being able to see many different labels or things, but I really could not tell you what those are because I really live within the biology sphere myself. And a big question that we want to know is whether or not we'll ever be able to truly do Ibex in 3 dimensions. And so I've heard many people, I've heard from Jose and Joao about trying to embed tissues inside of things to keep them from wiggling around and keep them pretty sturdy, and whether or not we can, try and do a whole volume with many labels. Although the amount that I suppose we'll be paying in microscopy time would probably get pretty high, but I'm willing to pay it. And then so that's everything. This is the group. There's so many people that work in the Jordana Wasserman group and in the Schroeder Institute, but I've only put the imaging people up on here and our technicians. I cannot thank CALM enough. And, I've had a great opportunity to interact with Chien and Ray very recently, and they're both lovely people. Andrea has supported us incredibly, and Ziv is the computation person at the NIH. And these are the beginnings of our Ibex collaborative team that we're trying to build and pursue here at McMaster. And with that, I'm happy to take any questions.
That's stimulating thought. So we'll open the questions either remotely or here. Great.
So the pipeline is very front end. It's really dependent on the quality of the antibody.
Yes.
So what you are doing to make sure you validate that.
It's extensive, in fact. So I'm in charge for the global Ibex community for thinking about what our validation strategy is going to be. If you have suggestions, very happy to hear them. As I am only content when we knock out validate in mice. I'm not happy with other things, but sometimes you knock a gene out and it kills a mouse, and the mouse can't live. So we're talking about doing things like co-expression to be able to prove that this is working. In humans, it's a lot more complicated because you can't really, you're not going to find very many knockout human. But a lot of the times we do pathologist validation, and then other times we'll do co-expression validation as well. But it's a very, very good question. And I think that really is where we're trying to go with the validated panels, where we can say, this is a published panel, that we know that everything in here is labeling the thing it's supposed to label. I mean, it doesn't need to be perfect to the point where there won't be any background in the system, because we are looking, not at single cells, but at tissues. But we want it to be at a point where we can feel very comfortable that the true signal is detectable above noise.
There's a large project on
Mm-hmm. Good. Good.
And it's been very surprising how many antibodies that are commonly used in research fields.
Mm-hmm. That's very good to know. And I think it's something that is in my mind a lot as we're looking at these things. So it's easier in the mouse. I'm sure you're talking about human for. That's very useful. I think also spatial transcriptomics will start to help us a little bit in this, that we'll be able to say, we can co-localize protein and transcriptional information. But thank you for the warning. That'll be something we'll have to keep an eye on. Joe, sorry. I apologize.
Sorry. Josh, that was a great talk. Thanks very much. I'm not a biologist, so I'm not the biologist side of things, but if I understand correctly, you do an image with 5 or 6 guys, then you use sodium borohydride to reduce
lithium. Yeah. Then
you apply the next set of antibodies.
Correct.
So, right?
Yep.
So how do you know that the lithium borohydride isn't doing anything to the biology?
It does. It does in some contexts. These tissues are fixed before we're imaging them. So really the cells are not alive, that the biology in of itself won't change. But what does happen is that lithium borohydride, some of the epitopes for these antibodies are sensitive to lithium borohydride. So there's actually, I think I have Yeah, so there's a process for the validating of these antibodies where when you get to this full panel or actually, even in the antibody validation step here, this is from like a broad consortium. But this here, when you do this for Ibex, you have to test both the reagent sensitivity for these antibodies and the bleach sensitivity. And some of them are bleach sensitive, but you can move those into the first panel of your imaging and then still be able to use those. So I think, if you end up getting a lot of reagents that are like that, that might end up becoming a bit of a question mark. But so far, I'd say 95+% are not sensitive to lithium borohydride. The other thing that's interesting is that many groups before Ibex were using peroxide to reduce, and it rips through the tissues. It destroys the tissues like very badly, and lithium borohydride for some reason doesn't do that in the same way. So very good question.
Does the tissue need to be sucrose-granule fixed, frozen, or can one use FFPE tissues?
So that's a very, very good question. You can use FFPE tissues. The process of the bleaching and the restaining for what makes up the Ibex portion of this really, any fluorophore that can be inactivated will be inactivated, and you can restain. The problem with FFPE tends to be that you mask all your epitopes with the paraffin embedding and dehydrating. So we don't suggest FFPE for a lot of the stuff we do, actually everything we do. And all of our validation ourselves is done in fixed frozen conditions. Now, the global Ibex consortium has many people who are thinking about FFPE because of historical samples and block embedded samples and things like this. And so there's a lot of minds on that exact issue. But for us and the stuff we're doing here, well, look, if you want to give us a student to work on that, hey, very, very happy to. But we're getting new tissues and freezing those down, and that's how we're doing things.
I have a question for my school biology background. So all that you mentioned is very nice and very descriptive. But what tools are the immunologists actually trying to come up with to modulate these systems? For example, like overexpressing it on a cell at a certain time point in development.
Yeah. I mean, 100%. Immunology is the land of generating a knockout mouse and then trying to understand how that works. And I think that doing these Ibex imaging experiments with a control group and an experimental group is fully within reach. So you can say, we're going to use a knockout mouse by comparison to a wild type mouse and see how the tissue biology changes in that context. I mean, there's likely. I think that it does tend to be very binary, at least in our field. But you can do things like transgenic expressions or single nucleotide polymorphism insertions using CRISPR. I mean, I've even seen some people who have been using CRISPR like screening and then going and finding those CRISPR inserts in imaging. There's a field around screening and CRISPR in that realm. So I think that really anything you can do by microscopy, you can do with Ibex. It just really comes down to the question you're asking.
Standardizing the workflow, have there ever been any kind of interlab studies that is the one when you report that?
Yeah, there are, yes, but there's a lot of work that's been done. So Andrea Radke works in Ron Germain's group at the NIH, and there's when they first started doing all this Ibex stuff, they went through and tested, I think, 30 different possible bleaching reagents and landed on lithium borohydride. Then they tested, I think, 10 or 15 different fixation conditions or something like that. I mean, Andrea's told me the numbers. I don't remember exactly what they are, but they've definitely gone through and iterated. And I think the idea behind the global Ibex community would be to continue iterating and try and find something that works out very well. There's a very complex issue that I don't know exactly how to deal with right now, but it turns out that different antibodies can be sensitive to the solutions that you use to stain them in. So if you put Triton into your antibody staining solution, some epitopes on some tissues are Triton sensitive. Take the Triton out, now you can see it. Some of them require Triton to be there. Some of them require Tween to be there. So I think there's going to be a lot of work trying to figure out the exact conditions. But for now, we've landed on the fixed frozen sucrose gradiented tool, just because that we have seen maximizes the number of epitopes that you can get. And I just think you have to start somewhere. So I mean, I'm very excited to see people continue doing that. And I think HubMap is trying to do stuff like that too.
My background is not really in biology, so don't mind my question. How likely is it? So when you're using these different dyes, Ibex, like different dyes, how likely is it that like same cell gets the same price?
Oh, always. So always. You know, so you start saying, how do you? So the way that you know that it's the same cell that you're looking at is that we actually align in X, Y, and Z space, and we recapture the exact same image that we were doing before. So we also run it through an alignment program called Simple ITK that will kind of fix the small differences. And then if that circle is there and that circle is there, you pretty well say that's the same cell. Now, you will always see multiple probes that will land on that cell. And in fact, you need that because that's how we describe the different pieces of the biological complexity. I suppose one question is, how do you know that the bleaching has worked? We used to image after every bleaching round. We don't anymore. It gets rid of the staining. I mean, DAPI will stay, Alexaflor 594 will stay. None of the other ones will, they'll all be gone. And you'll know pretty quickly too also if it didn't work, because you'll take a look and you'll see this weird pattern that, you've used these antibodies before and it shouldn't be like that. And you go, yeah, there was a problem. We've aborted a lot of Ibex runs.
Just going back to the bleaching stuff, why can't you just use white?
You can, except that some of these dyes are now too stable to be light photo bleached, which is good and also a problem. So, the thing is, the lithium borohydride will bleach in 5 minutes. If you use light, the PE, there's actually a whole tool, I think when it was on there, that you only stain with PE, and then you photo bleach the PE all the way out, and then you stain with another PE, then you photo bleach the PE all the way out. If you saw the region of interest that Joao got us on that last imaging run, I don't know if you could do that with PE. You'd have to be very patient. That's why we use lithium borohydride. Like Alexaflor 647 is incredibly stable. We've rerun some tissues like 30 times on it and it's still going.
With cells communication, are you going to use kind of network analysis to see all the connections?
Yeah. So yes, 100%. We want to. I think the computational tools is really the next big frontier for us, because now Vittoria is being able to go and get pretty highly multiplex tissues, and we need to stop just getting data and start actually making some learnings from these, right? Pathway analysis, which for those of you who don't know, is a way of kind of looking at for example, receptor ligand pairs and being able to predict based on those things which cells may or may not be interacting and stuff like that. And you can do that in huge dimensional space, and you can do that over hundreds of thousands of cells. And I've done this in our single cell RNA sequencing data set, and it's amazing and it's a lot, and you have to figure out what you're going to take from it. But it's I think it's really, really exciting. And so I do a little bit of computation myself, but honestly, I've been a little afraid of the imaging side of it. So I'm hoping Andrew will help us a lot with that one. Yeah. And if Wednesdays doesn't work, I can find some other times as well. And also, I've been told to say like and subscribe the BIMR page. So please do that. And when the Ibex community, global community stuff goes online, I'll make sure that it's available through CALM. And not going to be strangers. I talk to these guys every single week, and we're going to keep doing that. And I hope that a lot of us can live within CALM, and we can continue to build it out.
more biohazardous, things like that?
That's a really good question. We use it for BSL too. You know, I believe that formalin fixation is, it's basically 1% formaldehyde. 3 facility. Can infect with fluorescent viruses and chase. Cool. Yeah. Sorry.
All right, no, with that, let's thank Josh for a great talk and a great discussion.
Thank you.
Promise we did not pick him to advertise more.
And I will do it shamelessly every time.
And also remind you that next week we have the fourth seminar series with CALM BIMR seminar. It's going to be Dr. Angela Bardo, who's coming from UTEL, and we'll be talking about 3D printing at the nanoscale and biology.
We'll have a reception.
And we'll have a reception afterwards. Okay. So thanks everyone.